Hello and welcome to the third video on the GCSE biology revision section. So this is looking at cell transport. The specification reference is 4.1.3. So we're going to be looking at diffusion, osmosis, active transport, and then that required practical using potato and sugar solution, or any plant or vegetable and any kind of solution. So we'll start off by looking at diffusion. So diffusion, our official definition is diffusion is the spreading out of particles in a solution or a gas, resulting in a net movement from an area of higher concentration to an area of lower concentration. Now diffusion is a passive process, means it doesn't take any energy for it to happen. Cells don't need to use any energy to diffuse substances across them. All they need is a concentration gradient. There are several factors affecting diffusion, however. We've got the temperature. So the higher the temperature, the faster the particles are moving. They've got more kinetic energy, therefore their velocity is faster. If their velocity is faster, they will be moving around quicker, and as such diffusion will happen quicker. We'll get that net movement happening faster. Also, if we've got a larger concentration gradient, the higher the concentration gradient, so we have a large concentration versus a very, very small concentration, diffusion will happen quickly down that as well. If the concentration gradient is pretty flat, so the concentration of our high and low are pretty similar, diffusion will happen very, very slowly. So we have a big concentration gradient, diffusion will happen quicker. And then surface area as well. That surface area is a very, very important thing for it to for diffusion to happen. If we've got a large surface area, diffusion can happen quicker, especially when we look at our surface area compared to our volume, or what is known as our surface area to volume ratio. Thing we might need to work out on, you've just seen it pop up on your screen, so we need to be able to calculate surface area to volume ratio and compare them. The trick is to find the surface area of your object and then find your volume, get them. So we've got our surface area to volume. If you can simplify them down, get it to its in maths I assume that's sort of the lowest common factors. I think it's probably what they call it. Hopefully no math teachers are watching this and will criticize me, but making it as simple as we can. So please have a go at looking at the surface area to volume ratio for cube 1 and for cube 2. The answer will be on the next page, so feel free to pause the video now and have a go at working it out yourself. So let's start off by looking at the surface area of cube 1, and then we'll work out the volume and find the surface area to volume ratio. So the surface area is all the surfaces that are exposed added up together. To find the surface area of a cube, we'll find the area of one of the sides and then times it by 6, because there are 6 sides. So to find the area, we'll do 1 times 1, that's the height, times the width. 1 times 1 is 1. It's 1 centimetre cubed. We've got 6 faces, so 1 times 6 is 6 centimetres squared. So 6 centimetres squared is our surface area for cube 1. We'll now find the volume. So the volume is the height times the width times the depth. Okay, so that is 1 times 1 times 1, which gives us 1. So it is 1 centimetre cubed. So we've got our surface area is 6 centimetres cubed, our volume is 1 centimetre cubed. It's quite an easy ratio to work out because we've got it as simple as it will go. So our surface area to volume ratio is 6 to 1. So 6 for our surface area, 1 for our volume. It's quite a large surface to area to volume ratio. Looking at cube 2, our surface area, so same method, we'll work out the area of one face and times it by 6. So 2 times 2 is 4 centimetres squared. We'll times that by 6 and that gives us 24 centimetres squared. So our surface area of cube 2 is 24 centimeters squared. Working out our volume, we'll do the height times the width times the depth, and that is 2 times 2 times 2. So 2 times 2 is 4, times that by 2 again is 8, gives us 8 centimeters cubed for our volume. So our surface area to volume ratio could be 24 to 8, but obviously we can simplify that down as 3 to 1. If we compare them, we can see that cube 1 has a much higher surface area to volume ratio than cube 2, 6 to 1 compared to 3 to 1. So this is very, very useful for those single cells, things like amoebas and very small organisms such as insects and spiders, woodlice. They're able to get oxygen into their cells through diffusion. They don't need to use a respiratory system. They've not got lungs such as humans have or other mammals. For insects, they have something called spiracles. That's more of an A-level thing we'll look at. It means they don't need those respiratory systems for gas exchange. They can just rely on diffusion. They've got a very, very large surface area to volume ratio. You might notice this is becoming a key thing in biology. That surface area to volume ratio comes up an awful lot, so make sure you know how to calculate surface area and volume ratio. So at times when this comes in useful, we've got this large surface area to volume. We've got the alveoli in the lungs, those little air sacs in the lungs. They are very, very big. That means they can absorb oxygen and carbon dioxide and diffuse across them very, very easily. Gills on fish do the same sort of thing. They've got a very, very large surface area. They can get as much oxygen out of the water as possible. And then roots on a plant, you know from looking at your root hair cells, they've got a large surface area that increases the surface area available for diffusion. And it means diffusion takes place efficiently. So next we're going to look at osmosis. Now our definition of osmosis is the movement of water molecules from an area of high concentration to an area of low concentration across a semi-permeable membrane. Could replace that with partially permeable membrane, either is fine. So that is our definition of it. To do this, you need to know about percentage calculations as well, calculating percentage gain or loss, and you need to be able to plot, draw and interpret graphs. With osmosis, generally just knowing the definition, knowing that it's the movement of water, it's actually diffusion with water and it's going across that semi-permeable membrane. Again, it's a passive process, no energy is needed. In the exams, the times you might be asked about it are either as part of a larger question where we're looking at active transport and osmosis, for example, how to make stoma open with those guard cells, or you may have to do the required practical, which will assume that you know how to do calculations, so percentage calculations for loss and gain, also being able to draw graphs and interpret those. So you might remember there is a required practical. So this is where you're going to investigate a range of concentrations of salt and sugar solutions on the mass of a plant tissue. We normally do it with a potato in school because potatoes are quite easy to find and show a very, very good result, but you could do it with any kind of plant. You can have a look and you can see what is going on. So I would like you to write a method of how to perform this investigation. See if you can remember what to do for it. Give it a go. What I've done, I've put down a list of possible equipment that you may want to use. You may not want to use all of it, or you may want to. There may be other things you want to use as well. I'd say take about 6 or 7 minutes to try and write down as much as you can remember about doing the practical. Make it a good method. If you want, you can suggest which variables you're going to control while you're controlling them, what results you might find as well. What I'll do, pause the video now and you can have a go at doing it, and then we will look at the answers. So welcome back, hopefully you've paused it. We'll go on to the answers now. So this is the method that I have written up. So we're going to use a cork borer to cut 5 potato cylinders, so they're going to be the same diameter, and we're going to trim them so they're all the same length. We're making sure they are all the same to start with. We're then going to measure the mass of each potato and record it in a table. It's always important to write down your results. We're then going to measure the length of each potato and record it in a table as well. Again, making sure we are taking all these things into account. We're controlling as much as we can. In this case, I've chosen to look at sugar. Obviously, if the question was asking me to look at salt solutions, I would change sugar for salt. I'm going to have concentrations between 0 millimoles per litre and 1 millimole per litre. Obviously, for that 0, that's just going to be water. I'll just have distilled water. And when I get to the 1, it's going to be my stronger solutions. I can use that one solution to make up the rest of them if I need to. I'm going to put the solutions into test tubes, so each one into a different test tube, and I'm going to label them clearly with what each one is. I'm then going to place one potato cylinder into each solution, so each one now has one in. I've got 5 solutions, 5 bits of potato, 1 in each one. I'm then going to leave it for a certain amount of time. 24 hours is probably quite a good time. Any longer, we start seeing a few changes. If we do it too quickly, we don't see any changes at all. I'm then going to remove the potatoes from the test tubes one at a time. I don't want to take them all out and get them mixed up. I need to know which one is which. I'm going to blot them dry, and then I'm going to measure and record the mass and length of each potato and record it in my table. I can then compare the percentage change in mass and length of each potato, see what has gone on. If one hasn't changed at all, so there's been no change, I can then assume that the sugar solution it was in is closest to the strength of the sugar inside the potato. So the water had nowhere to go. There was no difference in concentration. What I will then do is draw a graph, have a look at my results, see where the change was, and I can work out the concentration of sugar inside a potato. Roughly. Obviously there are other things in there as well. I'm just looking at sugar, but it will give me a rough idea of how much sugar is in an amount of potato. I can compare it to my graph. It's going to be the point where there is the smallest change possible. So that is our osmosis required practical. Sorry, that was very, very badly explained. I will try and do a video of it demonstrating it as well. I always find that watching the practicals or doing the practicals is the best way to remember how to do it. If you are in doubt or you haven't got the time because of school being closed down, you can go onto the AQA website. If you search for the practical handbook, it will have all of the practicals on there and the methods that you will need to do, certain questions that you might want to have a go at answering as well. The last thing we will look at is active transport. So this is the last part of transport within cells. So active transport moves substances from a dilute solution to a more concentrated solution. So you may notice that this is different to osmosis and diffusion. We're going against the concentration gradient this time. To do this, we need energy. So we get that energy from respiration. Respiration happens in the mitochondria. So cells that have to do a lot of active transport will contain a lot of mitochondria, so they can make enough energy to perform that active transport. Now there are certain cells in the bodies or in organisms, so in plants and in animals, that will do active transport more than others. So in plants, it's really, really common in those root cells. So the root hair cells, they do diffusion for getting water in, or for getting certain ions in, and osmosis for getting water in. It's a concentration gradient, it's just going up. There are certain ions that there won't be many of in the soil, and there will be more of in the plant, so they will want to get them in because they need them for growth, and they need them to develop, especially chlorophyll, or magnesium for chlorophyll, the nitrates, so that's healthy leaf growth. They will get them in through active transport. They will expend energy making sure they get them in. That's how important they are for them. With animals, so we can get sugar from the intestinal tract. So inside our intestines, we've got a very, very large surface area to volume ratio on our intestines, lots and lots of villi, increasing that surface area. We want to try and get as much from the intestines into our blood as possible so we can take it to our cells. With sugar, when we have a large sugary meal, our blood sugar concentrations will be high. They will go down, obviously, with insulin, but right by the intestines, they will be very, very high. Sugar won't be wanting to diffuse across, so we will need to use active transport to physically remove it from the intestines and bring it into our bloodstream so we can take it round. We need to use active transport for that. It uses a lot of energy, but it is worth it. So, things you may need to do with transport in cells. You need to describe how substances move through or across cells, either through diffusion, active transport, and osmosis. So remembering that diffusion and osmosis are going down a concentration gradient, active transport is going up a concentration gradient. Because of this, diffusion and osmosis are passive processes, but active transport is an active process. It requires energy. Okay, so that's it for this video. I hope you find it informative and useful. We will be looking at organization next, so looking at tissues, cells, the heart, various things like that in the next series of videos. Please click subscribe. Thank you very much. Bye bye.